Hello, my name is Mr. Chipman, and I am the biology and AP biology teacher at Murray High School in Murray, Kentucky. And we are looking at Unit 0, Science Practices. Today, looking specifically at experimental procedures. What is an experiment? How does it work? All those things. Let's do it. So, a couple things about experimental procedure. You will definitely see these types of questions on the exam, so it's very important. A lot of this is going to feel like review, but it's interesting that it's review, and students still end up getting things mixed up over and over again. So, big thing with an experiment, there has to be a testable question, right? Question, is the sky blue, is not really testable, right? And so it has to be something that is actually testable. Look at the picture here. There's does water cause seeds to sprout? That is a testable thing, right? And so very, very important. The question has to be able to be tested, and those tests have to have something that's measurable at the end of that. So very, very important. Sometimes you will be asked to evaluate an experiment, whether or not it is testable or whether or not it is valid, and that is a proper thing. Can you test this experiment? Are you able to do that? And so with that come this idea of hypotheses. A hypothesis is an answer to a question that is posed, a testable question, and is, we talked a little bit about this in 0.1, and it is a possible answer for that, and then you test to see whether or not that is correct. And a prediction followed up with that is, if the hypothesis is true, then this is going to happen, right? If, acid rain, look at the example there on the right. Acid rain is killing fish. Well, if that is true and fish are dying in this lake, then this lake must be acidic, right? Can I test that? Sure. All right, and this goes back to the null hypothesis, alternative hypothesis. Remember, the null hypothesis assumes that the variable has no effect, right? That there's no effect on the actual system, whatever that system is, and that if there is an effect, if you can prove an effect, then you have to come up with an alternative hypothesis. You reject the null hypothesis, and you come up with an alternative one. You see there underneath null the idea of failing to reject. I think it's important to denote the difference between failing to reject, and accepting. The null hypothesis is a thing that you're setting out to prove wrong, right? And so you don't accept it, rather you fail to prove it wrong, right? You fail to reject it. So I think that's an important distinction, particularly as you are dealing with language concerning the null hypothesis. If you're asked to answer a question, you won't use that terminology accept, but rather say that you fail to reject that. We'll see that again with chi-square, which we'll talk about in 0.3. There are two groups in an experiment. There's the experimental group and the control group. The experimental group is the one that's being manipulated. You could also think of it as the group that has the independent variable applied to it. In this case, it's the pot that gets water. Right, and the other group is called the control group. The control group acts as a basis of comparison. Now, I don't have a particular slide for positive versus negative controls, but we can talk about it here, and that's fine. So there's two different controls. There's a positive control and there's a negative control, and there's a lot of confusion about them. And so I'll use this example here. Here you have two pots. One water is being applied to, the other is not being applied to. What is the purpose of that? It serves as the basis of comparison, right? If water is not applied, this is what happens. And the null would be that nothing's going to change. The water is not going to actually cause seeds to sprout. And because that happens, I have to have an alternative hypothesis, which is water causes the seeds to sprout, right? And so in this case, this is a negative control. In a negative control, there's no application to the control group. In a positive control is where there is a known application, right? So let's say that in another experiment, we apply water to one group and water plus fertilizer to a separate group, or maybe just fertilizer, right? Just fertilizer. We know, based on our previous experiment, what just applying water will do. It'll cause 9 out of 10 seeds to sprout. If we want all the seeds to sprout, maybe we'll try something that's unknown, right? And so a positive control is applying a variable with a known outcome. right? Versus applying no variable at all. Speaking of variables, let's talk about the variables. Control variables, these are things that are kept constant in the experiment. Notice this is different than a control group, which doesn't have the independent variable applied to it. The dependent variable and the independent variable, these are constantly mixed up by students. And I don't like the definition that I hear a lot of times, whereas the independent variable stands alone. Dependent variable depends on the independent variable. Okay. It's kind of a weak definition, right? Independent variable, this is the thing that is being manipulated in the experiment, right? Does it stand alone? Sure. You can't have more than one independent variable, but that is a weak definition. It doesn't really tell you what it's doing. If you go back up here and look at this experiment, water is the independent variable. It is what's being manipulated, right? I'm adding water to one, not adding it to the other, right? It does stand alone, I guess, in that it's the only independent variable. You can't have more than one independent variable because you can only test one thing, right? But manipulated, changed, that's a better definition. For dependent variable, measured. What is being measured in the experiment? What is the thing that you're measuring? It could be more than one dependent variable, not necessarily, depending on what your question is. Does the different light colors cause a difference in plant height? That doesn't have more than one independent or dependent variable. But maybe you just say plant growth in general, and then you measure the height and the width, and there's all these other things that you can measure. So anything that's being measured and can be plotted against the independent variable, that is going to be the dependent variable. A couple things here. Independent variables always found on the x-axis, no excuses, no exceptions, and dependent variable on the y-axis every single time. It's not a matter of, I want my graph to look different. Well, you just want it to be right. Independent variable x-axis, dependent variable y-axis. Moving on. And there are those things again, independent versus dependent. And so we've talked about this experiment. I'm going to move past this slide and talk about this idea of replication. All right. This is one that you might see on an FRQ question, and it will give you an experimental result and ask you if there's anything that could be changed about the experiment to make it better. And oftentimes it's going to have to do with numbers. So let's say that you have this experiment. I did an experiment with roly-polies, and I discovered that 50% chose the wet environment and 50% chose the dry environment. But I only had 2 roly-polies. If I replicated this, let's say I had 100 roly-polies, would I be able to replicate this and get these results? Maybe, but what's the chance of that happening as you get more and more sample size? It's less, right? And so a better experiment would be to do it with more on the front end, right? Having a larger sample size is going to give you more confidence in your numbers, and then those numbers can then be replicated later. And so concept of replication is very important in science, and we're not going to get into the importance of replication in science in general, other than to say for this class, a lot of times you will see this idea used where sample size tends to be the issue, or a weight of sample size, meaning that one group is getting more attention than another. And so that's another thing that you'll often see. Good experimentation has a large sample size and a consistent sample size as well. Very important. So let's look at some practice. A student designs an experiment to test whether caffeine affects the heart rate of Daphnia water fleas. Student places Daphnia in 3 solutions. You have just a plain water pond. You have a plain water pond with 1 milligram of caffeine and another with 3 milligrams of caffeine. All tanks have the same temperature, light, and oxygen levels. The heart rate of each Daphnia is measured after 5 minutes in the solution, which the following best describes the role of tank 1 in this experiment. So what's going on here? You have an experiment, and in the experiment we are manipulating the amount of caffeine given to Daphnia, right? And what are we testing? Heart rate. So heart rate is the thing that's being measured, which is the dependent variable. The amount of caffeine is the thing that's being manipulated, is the independent variable. In tank 1, there is no application of the independent variable, so it is acting as a control. And since there is no application, there's not a known variable being applied to it. This would be a negative control. Which one says that? Looks like A. It says that tank 1 serves as a negative control. Let's see if that's correct. Okay, it is. Forgot to ask you to pause. but that's okay. Hopefully this was helpful. And if you need help with chi-square, well, I'm going to be doing 0.3. Check it out. Later.